When researchers can view spatial relationships in X, Y, and Z dimensions, the understanding of the tissue microenvironment can spark new discoveries. This is the capability we are developing at ClearLight Biotechnologies. Our tissue analysis begins with clarity tissue clearing, followed by multiplexed immunohistochemistry. The stained tissue is then placed in a refractive, index-matching mounting solution for imaging with a confocal or light sheet microscope or another imaging platform. This allows us to perform our digital spatial image analysis. Imaging antibodies in 3D reveals novel insights unobtainable with other technology. We use confocal microscopy with field of view imaging. While we are not currently imaging the entire 5 mm diameter by 1 mm thick tissue sample, we are scanning the entire sample without image capture to identify any potential lesion or areas of interest. We then perform imaging in the proper field of view that contains the lesion, thereby not missing it during image selection. Researchers employ multiplex IHC staining to identify cell phenotypes of interest within the tissue microenvironment. Since the sample is embedded and intact within the clear hydrogel matrix, the same tissue has the potential to be imaged entirely in 3 dimensions. The tissue's transparency enables 3D imaging of subcellular components, such as DNA, RNA, and proteins for analysis within the tumor microenvironment of a tissue. Here we present a 3D model used to segment and classify cells in a tonsil. First, DAPI stains the cells in our tissues. Then our AI method can automatically process this information and represents each cell nuclei with a different color. You can see how PDL1 is used to automatically discriminate which of these cells are PDL1 positive and display them separately. You can then see pan-CK, which stains epithelial cells, and then finally, you see the CD8 marker. Altogether, this AI method provides the information required for a thorough characterization of the spatial distributions and morphological properties of all the cells within the image sample, as well as the classification of different cell types revealed by the biomarkers employed. Yesterday's science fiction is becoming science fact. It is now possible to trace the path of a single nerve projection through a forest of other cells. Researchers can illuminate the tissue microenvironment in a 3D volume. The see-through tissue clearing process can be integrated into a normal tissue processing workflow and be complementary with current 2D FFPE technologies to further empower researchers involved in preclinical and clinical research studies to see more biology.